Okay, so we've talked a lot about DNA and we've talked a lot about proteins. We had to kind of detour a bit to talk about proteins and also enzymes. And what I wanted to impress on you is that you make well over 20,000 different proteins, including enzymes, but also a lot of non-enzyme proteins in every one of your functional cells. And they all have to be made to the right shape if they're going to work. And if they don't work, you could have consequences that could range from minor to completely lethal, because proteins do so much. To live, you have to be able to make many thousands of different kinds of proteins by putting amino acids together for all of them in exactly the right order. And if you don't, you make proteins that don't have the correct shape. And if that happens, you may not, that may not be viable. So what's the link? Go back to the early 1900s. There's a British doctor who's studying a genetic disease called alkaptonuria. His name's Archibald Garrett. And if you have alkaptonuria, your urine contains high levels of a compound called homogentisic acid. And homogentisic acid is transparent when it comes out of you, but on exposure to air, homogentisic acid turns black. So that's normal urine on the left. On the right, that is a urine sample from someone with alkaptonuria. The sample has sat out for about 30 minutes, and you'll notice that the urine has turned completely black. Okay, just checking the chat to make sure I'm not missing anything. So Archibald Garrett is studying people with alkaptonuria. It's a little more serious than just having pee that turns black, because your body makes lots of homogentisic acid. Your kidneys get rid of as much as they can through the urine, but they can't get it all. And homogentisic acid will build up in the skin. Here's the ear of someone with alkaptonuria, and you can see that the skin has turned, starting to turn black. It can build up in the eyes, the heart, and especially in your joints. And in your joints, it damages cartilage. So if you have alkaptonuria, you're pretty much guaranteed to develop severe arthritis, usually pretty young, usually by your 30s. So this is not a condition that you really want. Homogentisic acid is produced when cells break down an amino acid called tyrosine. Tyrosine is over here at the upper left. Right there is the amino group. There's a carbon atom stuck to two oxygens right there, so that's the carboxyl group. And the R group looks like this, and this thing that looks a little bit like a 220-volt plug is a symbol for a ring of carbons and hydrogens. So this is one of the 20 amino acids that you need to make proteins, but most people get much more tyrosine than they need. The excess can be disposed of, and tyrosine gets broken by several steps. I'm not detailing all of them out, but it eventually is broken down into homogentisic acid. And then the homogentisic acid is broken down into this thing at the lower right. It's called 4-mallyl acetoacetate. Don't memorize these names, by the way, and certainly don't try to memorize the formulas. And then 4-mallyl acetate is broken down further and ultimately can be used as a source of energy. So this is how you break down and recycle the extra tyrosine in your diet, because most people get more than necessary. However, for homogentisic acid to turn into 4-mallyl acetoacetate, this step right there that has an arrow, there has to be present an enzyme called HGD, short for homogentisate dioxygenase. Now, remember, you can think of metabolism as being like very complex assembly lines, or sometimes disassembly lines, where every step is governed by one enzyme. If you block that step, you end up with, you can block the whole process. So if there's an enzyme called HGD that's missing, or if it doesn't work, this pathway is blocked and homogentisic acid builds up in the body. Your extra tyrosine gets broken down to homogentisic acid, but the homogentisic acid can't go anywhere because you don't have a functioning enzyme to break it down into formaldehyde acetoacetate. And so with nowhere to go, homogentisic acid builds up and ends up turning your pee black, and also begins doing damage to your cartilage. Garrod studied families that had a history of alkaptonuria, because that does tend to run in families. And he discovered that it was inherited as a simple Mendelian recessive allele. If you have 2 copies of the dominant allele, you don't have alkaptonuria. If you have 2 copies of the recessive allele, you have alkaptonuria. If you are heterozygous, you don't have alkaptonuria, but you could pass it on to the next generation. Alkaptonuria follows the same rules exactly as white flower color in Mendel's peas. Garrod studied several other traits like this that tended to run in families. And he concluded that what a gene fundamentally was the instructions for making one enzyme. As he put it, one gene, one enzyme. That's the link. That's the connection between all of this genetics and DNA stuff we've been talking about and the proteins. What a gene is the instructions for making one enzyme. We would actually expand this now and say one gene, one protein, because there are lots of proteins in your body that aren't enzymes, but nonetheless, they still do, they're still made using information stored in the genes. Okay, incidentally, I was talking about how your body breaks down alcohol on Wednesday. And I mentioned that you have one step that converts your alcohol into acetaldehyde, and then a second step that converts the acetaldehyde into acetic acid. And if you block that second step, acetaldehyde builds up in the body, and you get some of the symptoms of a hangover. There are some people that inherit alleles for a form of aldehyde oxidase that's less effective, and they are intrinsically intolerant to alcohol. This gentleman in this picture on the left, that's him before drinking a little alcohol, and that's him on the right 5 minutes afterwards. One common symptom is that 5 minutes after drinking, the skin flushes, turns red. So we talked about those drugs and those mushrooms that block aldehyde oxidase and make acetaldehyde build up and make you feel like you've got the hangover from hell before you've even gotten drunk. But there are some people that naturally inherit an ineffective form of aldehyde oxidase, and they'll feel terrible before they've gotten drunk all the time. Another example that you might have heard of, this is there a way for this guy to have a higher tolerance? There may be people that are born with more effective alleles of aldehyde oxidase who could drink a lot more before feeling hungover, or maybe never strongly feel the effects at all. If there are such people, I just want you to know I hate them. Just kidding. I'm not sure what can be done for the person with ineffective aldehyde oxidase. Well, there are types of gene therapy you could maybe try, but I doubt it would be worth it considering the risk. And that's really getting ahead of myself. This is a diagram of an enzyme called phenylalanine hydroxylase. And you can see it's got a complicated shape. It's got a certain primary structure. Its secondary structure you can see because it's got alpha helices in blue, or the one I circled. And there's a beta sheet area, this thing that looks like an arrow. And right in there is the active site that's got a molecule of the amino acid phenylalanine in it. And phenylalanine hydroxylase normally converts phenylalanine to the amino acid tyrosine. If your body can't make this, if it can't make phenylalanine hydroxylase, then if you eat phenylalanine in your diet, the phenylalanine builds up because you've got no way to break it down. And excess phenylalanine turns out to be toxic to the developing nervous system. Phenylalanine builds up in the body because the enzyme that would dispose of the extra isn't there. And you have a condition called phenylketonuria, or a PKU. And the reason that matters is if you don't catch it when you're a newborn, that causes developmental delays. That causes mental retardation. Fortunately, there's a very easy test for it, and babies are tested for it on their first day. All you need is a urine sample and some reagents that you pour onto the first wet diaper. And if they turn black, if that first wet diaper turns black when you add certain chemicals to it, the kid may have phenylketonuria. If you have phenylketonuria, you can live normal lives, but you have to avoid foods with high levels of phenylalanine. There's probably no way to avoid it completely. And in fact, you need some. It is one of the 9 essentials, but you can't take in too much. And this is why there's a warning label on bottles and cans of Diet Coke and other diet sodas if they contain aspartame, NutraSweet, because aspartame is partly made of phenylalanine. And PKU is also inherited as a simple Mendelian trait, following the same rules as white flour color in peas. So we now know that what a gene is it's responsible for making one polypeptide chain of amino acids. And the instructions for putting amino acids together in order are encoded in the order that the DNA bases are in. The order you put your DNA bases in determines the order that you put your amino acids in when you're making a protein. And that order of amino acids determine the shape that it's got. And this way of coding information in a piece of DNA is what we refer to as the genetic code. So how do we crack the code? Well, here's the thing. In a eukaryote, DNA stays in the nucleus, and it never leaves unless the nucleus breaks open. And if that happens, the cell is dead or dying. Normally, DNA never gets out of the nucleus, but we know that proteins are made outside the nucleus, not in it. And so we have to figure out how the information gets from the nucleus to the outside, and how do we read that information anyway? Well, Francis Crick, one of the co-discoverers of that double helix shape, proposed what he called the adapter hypothesis, that information is copied from the DNA onto a second molecule, and that second molecule leaves the nucleus and goes to assemble the amino acids into the right protein. Think of it like this. Imagine you're in UCA's library You know, they've got the Starbucks and a bunch of computers, and oh, they've also got some books. And you're poking around among the cookbook collection. They do have a few, and you find a recipe you actually want to try. Unfortunately, you can't take the book out of the library because maybe you've got some fines or something like that. So I guess these days you could just take a picture of the page with your cell phone, but pretend for a moment that that's not an option. What you could do is leave the book in the library, but make a photocopy of the recipe and then carry the photocopy out of the library to your kitchen and then cook the actual recipe. Or heck, if you didn't want to use the photocopier, if you're really old-fashioned, you could get the book, write down on pencil and paper the recipe that you want, and carry that piece of paper out of the library off to your kitchen where you make the actual food. Something very similar is going on here. DNA stays in the nucleus and can't leave it, but somehow the information that's carried by the order of bases is copied, and the copy leaves the nucleus and goes out to where proteins are being assembled. And Crick's hypothesis turned out to be right, and it turns out that the adapter, the molecule on which DNA information is copied, is the second major type of nucleic acid. It's ribonucleic acid, or RNA. And RNA is like DNA. It's a polymer of nucleotides, but there's 5 major differences. RNA is single-stranded. It's not double-stranded, and it doesn't usually form a helix. You will sometimes see a single strand might actually do kind of a hairpin loop and form hydrogen bonds with itself, kind of like a piece of tape sticking to itself when you're just trying to wrap a Christmas present. But it doesn't form the long double helixes that DNA does. RNA doesn't usually form a helix at all. RNA contains a different sugar. It doesn't contain the sugar deoxyribose. It contains the sugar ribose. And RNA uses one different base. It uses adenine, guanine, and cytosine, just like DNA, but instead of thymine, it uses a base called uracil, not Duracell, that's a battery. This is uracil, which we abbreviate U. And then finally, healthy eukaryotic cells, the RNA is found outside the nucleus. DNA is never is. So on the left, this is a DNA nucleotide that contains the sugar deoxyribose. And it's deoxyribose because if it was ribose, it would have another oxygen stuck to it with a little hydrogen added for flavor. If it was ribose, it would have that. But since it is not, there we go, it's deoxyribose. And the base up here is called thymine, shaded here in yellow. Compare that to an RNA nucleotide, which the sugar is ribose because it does have that oxygen and hydrogen right there. And the base that it uses is not thymine, it's uracil. You can see it's very similar in shape to thymine, it's just a little bit different. Okay, so RNA is the reason proteins can leave the nucleus. Proteins are made outside of the nucleus. There are some proteins that will go in to the nucleus. There's many that don't, but the place where proteins are assembled is outside the nucleus. And it's the RNA that is made in the nucleus, but leaves the nucleus and goes outside to do its job. Okay, so what happens here? In the nucleus, when a cell prepares to make a particular protein, one of the lengths of DNA unzips. And this is like what happens when DNA replicates, but in this case, RNA nucleotides are assembled to make a complementary strand to one of the DNA strands. And this follows the old familiar rules. Wherever there's an A on the DNA strand, the RNA strand will put a U, not a T. Wherever there's a T on the DNA strand, the RNA strand will have an A. Wherever there's a G on the DNA strand, the RNA will have a C. And wherever there's a C on the DNA strand, the RNA has a G. And when the RNA strand is completed, it peels off and the two DNA strands come back together and form that old double helix. So we've made a piece of DNA that carries information because the order of nucleotides, okay, sorry, backtrack. We've made a piece of RNA that carries information because the order of its nucleotides is complementary to one of the strands of DNA. That RNA strand is called messenger RNA, or just mRNA for short, and that's what leaves the nucleus and goes out into the cell. By the way, it's recently become possible to harness this. Two of the COVID vaccines, Pfizer and the one that isn't Johnson & Johnson, I'm suddenly drawing a blank, but the first two that were produced were actually mRNA vaccines. They're artificial messenger RNAs. They're not made in the nucleus of your cells. They're injected into your arm. But those messenger RNAs that are injected into you do the same thing as messenger RNA that's made in your cells does. And by the way, the process of making a complementary RNA strand to a length of DNA is called transcription. And there's a diagram of what it looks like. You can see right here, there's a double helix of DNA going from left to right. There's a big enzyme represented by this purple blob in the center. It's called RNA polymerase, and it does the actual work of assembling RNA nucleotides into an RNA strand. So right there, you can see that the double helix is unwound. And right here, complementary to one of the DNA strands, you have an RNA strand that's being assembled because RNA nucleotides are coming in and getting put into place by RNA polymerase. Incidentally, that's a picture I took last October in UCA's Nature Reserve. That's a mushroom that was fairly common at a particular spot that I know where it grows. I'm not going to tell you. It's called Amanita bisparigera. The common name for this mushroom is the destroying angel. And it's called that because it's poisonous. And what the poisons do is block RNA polymerase. They stop transcription. A cell that has been dosed with Amanita poison can no longer carry out transcription. It can't make messenger RNAs, which means that it can't make proteins, which means that it starts dying. So if you eat Amanita bisparigera, this has the nasty effect of killing your cells, and the first cells that get killed tend to be the ones in your liver. So if you were to eat even part of this mushroom, your liver would start dying, which means you would die in a very unpleasant way over the course of about a week, unless you got an emergency liver transplant. So there, that's a natural substance that blocks transcription. And if you do that, the end result is a rather horrible death, a rather drawn out and painful death, I might add. Moral of the story, when you go on your field trip to the nature reserve, don't eat the mushrooms. There actually are some in there that are edible, and in fact, they're quite tasty. I am told that Amanita bisparigera actually tastes pretty good if you eat it. But yeah, you could use that to poison somebody. The most famous person who was deliberately poisoned by mushrooms was the Roman Emperor Claudius. He had one species that was his favorite, and it was quite delicious, but I think his wife bribed the cook to add some poisonous mushrooms into the mix with the delicious ones, and it killed it. That's why I'm not telling you where this grows. Okay, so assuming you have not eaten Amanita bisparigera, and assuming that it's not killing your brains like a poisonous mushroom, deadly. When I play, don't melt it. Okay, sorry. Don't know what came over me there. Something just got a hold of me tightly, flowing like a harpoon daily and nightly. The messenger RNA passes out of the nucleus if it's a eukaryote. If it's a prokaryote, all of this is all happening in pretty much the same space. But either way, the messenger RNA goes out to a tiny structure inside a cell called a ribosome. And there are tens of thousands, maybe hundreds of thousands of ribosomes in a typical cell. The ribosomes are way too small to see with a light microscope. There's a physical limit to the minimum size of things you can make out using light, and ribosomes are below that. We can see them with powerful electron microscopes, but you would never have seen these in lab. Each ribosome is made up of about 50 proteins stuck together with yet another type of RNA called ribosomal RNA. We're not going to worry too much about what it's made of. What you need to know is that a ribosome is made of two subunits, and in between the two, there's a groove. And the messenger RNA goes out of the nucleus. It moves to one of the million ribosomes, however many it is. And the messenger RNA makes like Madonna back in the early 80s and gets into the groove. Of course, not that you remember that song, because none of you were alive back then. Get into the groove, boy, you got to prove your love to me. Okay, never mind. That's a model of what a ribosome looks like that we've worked out using both powerful electron microscopy and also X-ray crystallography. It probably looks like a big pile of spaghetti. Don't worry about all of that. What I want you to notice is that we have one subunit right here that's shaded in kind of a purple and teal, and one subunit right here that's shaded in purple and gray. Wow, that's like UCA colors right there. And in between the two units, there is this groove. And messenger RNA slots into the groove, fits itself in. Okay, now what? The messenger RNA has gotten into the groove. Now what? There's a second type of RNA that you need to know about, and it's called transfer RNA or tRNA. And a tRNA molecule is a single strand, but it's twisted on itself to form a set of loops. One of the loops, called the anticodon loop, has 3 base pairs that happen to stick out in a prominent way. It's twisted in such a way that 3 of those bases, I should say bases, not base pairs, 3 of those bases are sticking out And one end of every transfer RNA is bound to an amino acid. And which amino acid is present depends on the bases in the loop. So that's a simplified model of a transfer RNA molecule. The actual ones look a little bit more twisty. It is a single strand, and it starts here and ends there. But the strand is base paired with itself, and the overall shape is kind of like a twisted clover leaf. And right down here is the anticodon loop, and there are 3 bases on the anticodon loop that happen to stick out, kind of like buck teeth. And then here at the end of the transfer RNA, an amino acid is stuck to the transfer RNA. And which amino acid it is depends on which bases make up that anticodon loop. Okay, so this is how we read the genetic code. Messenger RNA carries information to the ribosome, and the order of nucleotides in a messenger RNA molecule determines the order of amino acids in the protein. And the nucleotides are read not 1 at a time, but 3 at a time in groups of 3 that are called codons. And every codon is associated with 1 amino acid, and only 1 amino acid, except for 3 codons, which are called stop codons. They don't code for an amino acid. They're kind of like periods at the end of a sentence. What they do is tell the ribosome to stop here and stop assembling this protein. So here's a ribosome, drawn much more simply than that diagram that I made. And there's a piece of messenger RNA that is like that Beatles song. It comes grooving up slowly, and it slips into the groove on the ribosome. I haven't drawn the nucleotides over here. There is kind of a lead-in sequence that enables the ribosome to find where it's supposed to go. So there are more nucleotides over in this part that I'm just not showing. But the messenger RNA has slipped into that ribosomal groove. And you will notice that right here, we have 3 nucleotides that are just inside the groove on the ribosome, and they are AUG. Okay, there's a whole bunch of transfer RNAs that are floating around, and various ones will try to fit into the groove and form base pairs with the messenger RNA. The only one that can successfully do this is the one whose anticodon loop has UAC, because UAC is complementary to AUG. All of these others over here, if you try to fit them in, they'll fall off because their anticodon loops do not have UAC, so we don't get hydrogen bonding here. Again, you could think of it as many keys fitting into a lock, and the only key that will open the lock is the one that has teeth of the right shape. Here, those bases on the anticodon loop are acting kind of like the teeth on a key. And thanks to the work of special enzymes called aminoacyltransferases, transfer RNAs that have UAC on the anticodon loop always carry the amino acid methionine, which I've abbreviated as MET. That green circle with label MET represents the amino acid methionine. All of these other transfer RNAs are carrying different amino acids. So the only tRNAs that are complementary are the ones that are carrying methionine. Okay, now what? Now the messenger RNA goes ka-chunk, ka-chunk, ka-chunk. It moves 3 nucleotides down. And now the nucleotides in this position happen to be UUU. Well, all of these transfer RNAs will try to fit into that slot, and the only ones that can fit are the ones whose anticodon loop has AAA. That's the only ones, that's the anticodon nucleotides that are complementary to UUU. Transfer RNAs that are carrying AAA on the anticodon loop also carry an amino acid called phenylalanine. Okay, so the next three nucleotides are UUU. The only tRNA that matches UUU is the one that's carrying AAA. And transfer RNAs that have AAA also carry the amino acid phenylalanine. What now happens is a peptide bond forms between the methionine and the phenylalanine, and that first transfer RNA falls off, and it ultimately gets recycled. And the messenger RNA moves down another three nucleotides, click, 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 and the next three nucleotides in the ribosome happen to be UCA. Yes, our university is one of a small number of universities in this country that is also a messenger RNA codon. And all of the transfer RNAs try to fit. The only one that fits is the one whose anticodon loop is carrying AGU, because that's base pair complementary to UCA. And transfer RNAs that carry AGU on the anticodon loop also carry an amino acid called serine, S-E-R-I-N-E, serine. So yeah, if someone asks you what's UCA, you can tell them it's the messenger RNA codon for serine. Ooh, ah. So there, if UCA is right here, the only one that'll fit is the tRNA that has AGU and carries serine. And this process keeps on repeating. The ribosome keeps moving three nucleotides down. So what'll happen next is it will move down and UAU comes into the ribosome, and the only transfer RNA that fits is one that'll have AUA, and then it'll move down three more, and the next codon is GGG, and the only transfer RNA that fits will have CCC, and then it'll move down again, and the nucleotide CUU come into the codon, the ribosome, and the one that fits will be the transfer RNA GAA, and so on. And we keep on going up until we hit UAA, UAG, or UGA, which stops the process. And this happens to be the complete code book. On the left are the first position nucleotides, the seconds are at the top, and the third are on the right. So U stands for the amino acid phenylalanine. And let's see, ACA stands for the amino acid threonine. And GGG stands for the amino acid glycine, and so on. All of these three-letter codes that you see are just abbreviations for the 20 different amino acids. And again, you don't have to memorize what those codes are unless you decide you want to major in biochemistry. But that's what they represent. There is every one of the 20 amino acids has at least one codon that stands for it, and you'll notice that most of them have more than one. So phenylalanine can be UUU, UUC. Serine could be UCU, UCC, UCA, or UCG. That's the ones over here. Arginine could be CGU, CGC, CGA, CGG. So, excuse me, actually all of the amino acids except methionine, which is only AUG, all of the others have more than one codon that represents them. And then there's 3 codons up here called the stop codons. They are UAA, UAG, and UGA. And when the ribosome hits one of those, the ribosome falls off and the protein is finished. And we call this whole thing translation. So once the ribosome hits the stop codon, the completed protein falls off. It goes to wherever it's needed to go. We'll talk a little bit later as to what might happen to it. The protein might also have to be chemically modified before it can be used. There's lots of ways that can happen that don't concern us here. It's called post-translational modification, if you really must know. Woofda. So here we are at the top of the mountain, and I feel bad for everybody that couldn't join us, but let's break it down one more time. What a gene is a length of DNA, and that length of DNA carries the information needed to make one protein, or at least one polypeptide chain. The information that is needed is stored in the order of bases in the DNA. DNA has those bases arranged in an order, and the order is not random and it's not arbitrary. The order might just look like ATG, AAA, UAU, AT, AT, TTT, GC, GCG. You know, it might not look like much, but the order that those bases are in carries information, just like the order of letters in a sentence carries information. We use that information to put amino acids together in the right order to make a protein. And we have to put them together in the right order, or the protein may not have the right shape. And a protein that doesn't have the right shape can't do what it's supposed to do, or can't do it as effectively. Okay. So in order to put amino acids together in the right shape, we have to use the information that's stored in the DNA. And we read that information by a 2-step process. We first make a copy of it in the nucleus onto a piece of RNA, messenger RNA, in a process called transcription. The messenger RNA then goes out to a ribosome, and the messenger RNA's information is used to assemble amino acids in the right order, and we call that process translation. And there you have it. DNA is also inherited. We've talked about, you know, chromosomes. They're what you get from your parents. You know, you might say, I've got my father's eyes. Well, that's not literally true. At least I hope it's not true. I hope you don't have your father's eyes like in a jar under the bed or something like that. What it means is that what you got from your father is long pieces of DNA that carried information, and that information is used to assemble proteins. And some of those proteins contribute to your eye color, right? You didn't inherit your eyes from your dad or your mom or anybody. What you inherited was the information used to make eyes of a particular color. And that information is carried in DNA, and it's read by copying it onto messenger RNA and then reading it at the ribosome in order to assemble amino acids in the right order. And if you inherit flawed instructions for making a protein, you may end up with a genetic disease like alkaptonuria, because with faulty instructions, you can't assemble a protein out of amino acids in the correct order. And there we are at the mountain peak, and I will go ahead and stop the recording.